What drives me and what still drives me today is that I want to empower first responders with the right tool, which is going to streamline the diagnosis process. Because I have experienced firsthand how frustrating it is to not have the right tool, but still being treated and being in limbo without knowing what is happening.
Hello and welcome to the Blueprint Sessions. Today, I'm thrilled to introduce someone that I've had the honor of working side by side with on FluoretiQ's mission. I'm here to introduce Josephine, who's our co-founder, inventor, and CTO at FluoretiQ. I've had a front row seat at watching her turn what was a initially revolutionary idea into a tangible solution. It's a pleasure to have you with us today.
It's a pleasure to be here.
All right, so just a couple questions, to really start to share what the journey has been like. So having seen you build this company from the very start, I know that your journey has been unique. And so can you tell us a little bit about your decision to start this company and to occupy the CTO role and what that journey has been like?
Okay. So let's go from the beginning. Initially, you were part of QTech, which was a program that gave you the opportunity to translate your PhD into a product. Now, with this program, it meant that you had to travel quite a bit. And when you came to me, you said you cannot do all the traveling and still do the technical work. Now, after several conversations, in early 2017, I did decide to join as the CTO. And one of the first tasks I did was putting in sweat equity to gather feasibility data, which allowed us to apply for our first successful Innovate UK grant. Now, what drives me and what still drives me today is that I want to empower first responders with the right tool, which is going to streamline the diagnosis process. Because I have experienced firsthand how frustrating it is to not have the right tool, but still being treated and being in limbo without knowing what is happening.
Thank you. So what are some of the challenges that you've had to navigate in building this technology from scratch and translating it into a product? And how have your prior experiences shaped the way your approach to developing the solution?
So there are three major challenges. The first is credibility. Now, as a startup, nobody knows you. So we have to work very hard to generate evidence that we share with the world. But also, I need to balance, in my role, I need to balance the ability to shout out to the world what we are doing, but also protect the IP, which is a key value to the company. The second is managing a team. Now, our product is multidisciplinary. And with that, being an engineer, having to lead all these people, I had to invest a lot of personal time to speak the language. I'm not a chemist, so I need to be able to read enough to be able to relate with the chemists to support them. So that is quite challenging. And then finally, balancing my personal life. So I'm a wife and a mother to two beautiful children. I'm very fortunate that I'm well supported at home and within my community, but I cannot take that for granted. Now, through all of this, I always see the challenges as an opportunity to learn, an opportunity for growth.
That's excellent insight, and I don't know how you do it. Now, watching and observing you over the years, one thing has been clear to me, and that it's your approach to developing things is really dead set on collaboration. And as you pointed to having interdisciplinary teams, can you share a little bit about your experiences throughout the years of developing the products and how collaboration has helped?
So collaboration is one of the key things for me, especially in my role. In fact, I seldom turn down an opportunity to collaborate. I've had the privilege to collaborate with 7 educational systems. So just to name a few, because there are so many incredible people that I've worked with. So we've had the opportunity to work with the University of Bristol, with the chemistry department and the microbiology department. And we worked on our first Innovate grant where we looked at the early feasibility of using glycans to target bacteria in urines. We've also worked with Griffith University, which helped in continuing that investigation on how to use glycans to target bacteria. Other institutions we worked with include NPL, which is a National Physics Laboratory, STFC, Newton Gateway, and they helped us to confirm the surface chemistry on our microparticles and also confirm the adhesion mechanisms of the bacteria to our particles. In terms of labs, we've worked with Gateshead, Southmead, Gloucester Hospital, Axiom Labs, just to name a few. But again, as I said, we've worked with really incredible people.
That's excellent, and it underpins a real focus on collaborating wider with the industry. I'm sure it gave you lots of amazing insights.
Oh yes, it did. Absolutely did.
So I'm going to embarrass you a little bit because I know very early on you've had a personal experience with microbiology and urine testing, and I think it would be great to share the story of your personal experience with what's done today and how it influenced your approach to developing solutions for urine microbiology tests.
Okay, so I think the experience you're talking about was when we went to Gateshead pathology lab, and that was very early on in our journey. So they were kind enough to give us access to urines that were destined for waste. And so when we got there very early in the morning. It was a group of 3 of us. We got there and we had the opportunity to test over 300 measurements on urine samples. Now, this was my first time testing urines. The smell was horrible. And I thought, how do people sit in there day in, day out to do this work? But it was quite refreshing because we've never done that before. But sitting there and doing those experiments and also getting a laptop made me realize 2 things. The first thing was that the urine matrix is quite complex. Urine, I always thought, was just a yellow liquid that looks very nice and clear. But having to test this up close, some of the samples were cloudy, some were bloody, some had crystals, among many other things that I didn't know could be in urine. And so it made me realize that with all the development that we need to do, we have to put testing actual samples at the core of it. So right now, whatever we have to test, it doesn't matter how fragile the component is, we make sure we test with urine samples. The second point was, while going through the tour, they showed us this really well-equipped lab. It had all the latest tools that we could think of. And they will test over 600 samples in a day. They were open morning and night, and most of the week as well. So this was a lab that was running constantly. And at the end, they said, out of the 600 samples, majority of them were culture negative. Now, what that made me realize is that there are people out there that are on the UTI care pathway that might not need to be there, and it will take a couple of days to confirm that they don't need to be there. Now, I realize that this is inefficiencies, that it wasn't intentional, but there are inefficiencies in the system that gives the opportunity for what we are currently doing.
Wow. Yeah. And yeah, kudos to you for spending a full workday running through urines, but it's clear that it's given lots of useful insights into the problem space and what good might look like. So I think this is a great time for you to introduce, the technologies that you're working on and you've been developing at FluoretiQ.
So the Verify platform identifies the bacteria which is responsible for a UTI. And the way it does that is using probes that will specifically find the bacteria in a sample and report the presence of the bacteria.
Cool. And it does this all in just 15 minutes.
It does all in 15 minutes.
Amazing. So on the topic of speed, actually in the sector, it's sort of like a rule of thumb that usually, most developers have to trade off accuracy for speed. How did you tackle this with the Verify platform?
So fortunately, we didn't have to trade speed for accuracy or vice versa, because what we did do was very early on we defined a set of specifications that were informed by market insights, and created acceptance criteria which allowed us to assess if the specifications have been met. So, for example, for our probes, one of the criteria that we put is that any probe that we do put through into the pipeline needs to be growth-free and work directly from urine. In terms of reliability, we ensure that all components or prototypes that are created before we move to the next stage is tested with urines, because as I mentioned earlier, it is at the core of it, and we have to make sure that whatever we make is going to work in the field.
Amazing. So when we think about AMR, it's more than just a human health challenge, it's also
Yes, it is.
animal health. And one of, I guess, the accepted approach, or true approach to tackling this successfully is a One Health approach. From your perspective, what about the Verify platform makes it well, or does it line up well with this framework, and what makes it line up really well with the One Health framework?
I mean, first, for starters, pets also get UTIs. So it's not just a human problem, it is also a pet problem. And the way the Verify platform works is it identifies bacteria from liquid samples. So as long as there's bacteria in a liquid sample, it will identify it. Unfortunately, the pets also produce urines, which means that we can use Verify to test bacteria from the animal urines.
Okay, and how did you satisfy yourself that you wouldn't face any drop in performance or difficulty? Because I imagine, I'm not an expert on this, but I imagine cat and dog wee is probably really different from human wee. So how did you satisfy yourself that the platform will be ready for both?
We tested it with animal urines, as simple as that. So once we'd done the test in human urines, it was time to find the animal urines and repeat the test again. So we went through 3-4 months of just testing urines again, and another eye opener, and ensuring that we get the same results that we got with the human urines.
Amazing. Well, hats off to you again for doing that. Right now, I'd just like to change gears a little bit and talk about your experience as a leader. So you've had to lead interdisciplinary team. And what does it mean to you personally to be the one leading the technical team for delivering this solution?
It's an absolute privilege. I have a very curious mind, and I'm always thinking of ways to make things easier to use, more efficient. Sometimes I'm told I try to make things too efficient. And so with leading the team, I get a first row seat, seeing how brilliant minds can bring innovative solutions to the front row, to the market.
Yeah, and yeah, well done to you on making progress through the milestones. So I have just one question left for you. What animal do you think best represents you, and why?
I had to carefully think about this one. And the one I think represents me most is octopus.
Interesting.
Okay, bear with me. So it is very adaptable. So looking up the octopus is very adaptable and is resilient as well. And when I think about the octopus and its arms, I think about my job. I have to wear several hats in a day. Depending on which day and time you meet me, I could be somebody completely different in the company. So I feel like the octopus best represents my role and my personality.
All right, Josephine the octopus. Thank you very much for joining us on this Blueprint session, and thank you, Josephine, for making the time.
Thank you very much.
There are more sessions to come, and I hope you can join us for it. And if you found this video informative, please do like, share, and subscribe. And remember that getting the first prescription right makes a massive difference for our pets, for our planet, and for all of us as people. We'll see you next time.